Really excited to be participating in this panel with all of you with such great, amazing speakers. I have had a little bit of a peek at their talks, and it's going to be great, and we're going to have a great discussion afterwards. So everyone who's attending, we're asking you to put your questions in the Q&A box as we go through, and then we'll address those at the end. So I'll introduce each speaker while Joan cues up the videos, and so we'll transition through, and then we'll have our discussion at the end. So our first speaker is Dr. Ami ZOTA, who is an associate professor at the Department of Environmental and Occupational Health at the George Washington University Millikan Institute School of Public Health. She directs the ARISE EJ Lab at GW, whose mission is to secure environmental justice and improve health equity through advancements in science, policy, clinical practice, and communication. Her current research examines how socio-structural factors such as racism, classism, and sexism shape beauty product use, environmental chemical exposures, and the health of women and children. Ami is equally committed to science communication, and her work has been featured in high-impact media publications, including the Washington Post, USA Today, and the Atlantic Monthly, and has helped shape health and safety standards for consumer product chemicals. Dr. ZOTA is the founding director of Agents of Change in Environmental Justice program that empowers EJ leaders from historically excluded backgrounds in science.
Good afternoon. My name is Dr. Ami ZOTA. I'm an associate professor at the George Washington University Millikan Institute School of Public Health. Today, I'm going to talk to you about integrating intersectionality into the exposome, a novel approach to racial inequities in uterine leiomyoma. Before I dive into my research, I'm going to provide a brief overview. Uterine leiomyoma, or fibroids, are the most common tumor in women. They are non-cancer tumors of the uterus. They can be in various locations of the uterus as well as outside of the uterus. A woman can have one or many. They are smooth muscle in composition, and these fibroids arise out of the myometrium of the uterus. Estrogen and progesterone are crucial to the fibroid life cycle. Established risk factors include age, so fibroid risk and burden increases with age up until the time of menopause, at which point fibroids tend to decline, and as well as race, ethnicity. And we'll talk a bit more about that later in the presentation. Fibroids are associated with a large societal and economic impact. The majority of reproductive age women will have fibroids, although for many of them, they will go undetected and will remain asymptomatic. So 70% of US women are impacted, and over 80% of African American women will have fibroids. They're associated with substantial morbidity, including anemia, pelvic pain, pregnancy complications, fertility problems. Hysterectomy remains the only permanent intervention, and consequently, fibroids are the primary reason women get hysterectomies in this country. And the estimated annual cost have been estimated up to $34 billion. Want to return now to the disparities, the racial disparities. Fibroids are more severe and common in Black women compared to white women along multiple different axes. So they have a 3 times greater lifetime risk of experiencing fibroids, much higher risk of hospitalization and invasive procedures for their fibroids. And this is because fibroids are larger, more numerous, grow more rapidly, and occur at an earlier age in Black women compared to white women. I now want to talk about the conceptualization of race in fibroid research. Most fibroid scientists have conceptualized race as a biological factor and have focused on identifying molecular and genetic mechanisms responsible for racial disparities. Although studies have reported some biological differences between Black and white women with respect to their fibroids, a large recent study out of Harvard concluded that genetic and molecular factors do not explain the increased fibroid burden for Black women. It is really important to highlight race is not a biological construct. racial categories are weak proxies for genetic diversity, and use of biological concepts of race in biomedical and public health research is problematic because racial groups are heterogeneous and lack clear-cut genetic boundaries. Race is a social classification of people based on phenotype, and structural racism has been underexamined in fibroid research and in public health and medicine more generally. And when I talk about structural racism, I'm referring to the confluence of institutions, culture, history, ideology, and codified practices that generate and perpetuate inequity among racial and ethnic groups. Continental ancestry, migration, and correlated genetic variants are important, but these factors cannot be deduced from self-reported race and require more complex admixture mapping. So the question that I want us to grapple with is, how can population health sciences inform a new paradigm to help advance racial health equity in uterine fibroids? The ideas that I'm going to be presenting are coming, were published earlier this year in this commentary in the American Journal of Public Health. So I argue that we, to kind of more to better equip ourselves to handle racial health equity, we need to modify our existing model of the exposome. The exposome is a contextual model of disease that considers the totality of an individual's environmental exposures across the life course. It differs from traditional epidemiologic methods in three ways: expanded and dynamic exposure assessment. So it includes both these external measures of exposure, such as pollutants in air, water, food, consumer products, as well as internal measures of exposure. These are the biological responses to our external measures of exposure and are often measured on omics platforms, and such as the epigenome, the proteome, the metabolome. This high-dimensional big data is often used for data-driven discovery of unexpected exposure disease associations. Another framework that I think needs to be, and we propose for it to be integrated into the exposome framework, is intersectionality. Intersectionality is a critical theoretical framework that is really rooted in Black feminist scholarship. It was first coined by Kimberly Crenshaw to address the synergistic experiences of Black women who endure multiple forms of oppression as both Black and female. Some of the core tenets of intersectionality are that social identities are not independent and unidimensional, but multiple and intersecting. And we need to think about how social identities at the individual level intersect with interlocking systems of privilege and oppression at the macro social structural level. So how race intersects with racism, or how gender intersects with sexism. And that people from multiple historically oppressed and marginalized groups are the focal or starting point. So to help move us into a new way of addressing racial inequities in uterine fibroids, I started the FORGE study, so fibroids observational research on genes in the environment. So this is a transdisciplinary research effort that leverages expertise from environmental health, epidemiology, gynecology, epigenetics, and social psychology to address modifiable risk factors in uterine fibroids with an emphasis on racial inequities. So what I'm going to be presenting for the rest of my time here is kind of some proof of concept studies that I've published. All kind of the study population being premenopausal women undergoing surgery for fibroids at the GW Medical Center. And towards the last part of my presentation, I'll talk about kind of where we're moving next with the FORGE study. The two case studies that I'm going to talk about focus on phthalates, man-made chemicals used in hundreds of everyday products. Low molecular weight phthalates are most commonly used in personal care products as solvents to bind fragrances. High molecular weight phthalates are often used in polyvinyl chloride plastics to make them soft. There is quite a bit of data to support biological plausibility for phthalates as a risk factor for fibroids. I mentioned earlier that estrogen and progesterone are crucial to the fibroid life cycle. Phthalates can disrupt signaling pathways mediated by estrogen, androgen, as well as the PPAR activated receptors. DEHP is a reproductive toxicant in animals. So this is one particular toxic phthalate. And it has also been shown in experimental studies to be able to influence biological processes in fibroid cells, particularly those relevant to fibroid growth. And we're going to hear more about the epi literature, but one meta-analysis of several small cross-sectional case control studies found that DEHP exposure was significantly associated with increased fibroid risk. And this figure right here, which comes out of a review paper that I helped co-author last summer, shows the various mechanisms by which EDCs can affect the fibroid life cycle. So with this biological to context in mind, the first objective of our research was to examine the association between phthalate exposure and measures of uterine fibroid burden and severity. This is a cross-sectional study. We were interested in measures related to fibroid burden. So one such clinical measure is the size of the largest fibroid. It can be as small as 1 centimeter, can be as large as over 20 centimeters. We also looked at uterine volume, which we conceptualize as an aggregate measure of kind of the burden of all the fibroids combined. Since as the number and size of the fibroids grow, so does the volume. The uterus literally expands. Just a bit about our study population. So this is a small sample. Most women were Black, African American, overweight, privately insured, and highly educated. We also had variability in fibroid characteristics and surgical intervention. So we had women both who went through hysterectomies as well as myomectomies, which is kind of the selective removal of fibroids while keeping the uterus intact. In our study population, consistent with prior national studies, we saw exposure disparities. Notably, Black women compared to white women had higher exposures to beauty product-related phthalates, such as monoethyl phthalate metabolites. And this is consistent with my prior research around the environmental injustice of beauty and racialized beauty norms and beauty practices that inform these exposure disparities. We found that several high molecular weight phthalates, such as DEHP, were associated with increases in uterine volume, over 30% increase in uterine volume, but we did not see any associations with size of the largest fibroid. We also found that a potency-weighted sum of anti-androgenic phthalate metabolites was associated with an increase in uterine volume. So this was our way of looking at biologically relevant phthalate mixtures, as opposed to kind of more of the data-driven methods that others use. Our second case study went further to really try to understand mechanisms of action and to specifically look at whether phthalate exposures are associated with epigenetic modifications that are known to be relevant to fibroid growth. Here we focused on microRNAs. So these are small non-coding RNAs that regulate post-transcriptional gene expression by interacting with messenger RNA. We chose microRNAs because they help regulate processes central to fibroid pathogenesis, and they've also been shown to be influenced by environmental chemicals, at least in other target organs and tissue systems. This is the first study to specifically look at environmental determinants of microRNA expression in fibroid specifically. The prior studies kind of in the fibroid world have really focused on differential microRNA profiles between fibroids and adjacent non-diseased myometrium. And there's some experimental data that has actually found functional roles of some of these differentially expressed microRNAs. So what we did is we actually collected fibroid tumor tissues and myometrium from the women in our study who were undergoing surgical interventions. We used Tachman open array to quantify expression of 754 microRNAs. After multiple comparisons adjustment, we found that about 10% of the microRNAs varied in expression between fibroid and paired myometrium. So this was kind of our confirmation to be able to kind of look at the quality of our data and compare it with these other studies. We did not see, after multiple comparisons adjustment, any significant differences by race, ethnicity. We found a few significant phthalate microRNA associations. And very interestingly, I think this is one of the most interesting parts of this exploratory study, is we found quite a few of the phthalate microRNA associations varied by race, ethnicity, so that, you know, maybe only there was an association in one racial group and not the other, although the directions of the associations also varied. And, you know, so there are a lot of limitations of this research, and I think it's important to take a step back to talk about what can we glean from these proof of concept studies. So in our opinion, the results support hypothesis that phthalates may contribute to fibroid pathogenesis and epigenetic processes may play a role. The effect modification by race ethnicity suggests the need to look at social environmental interactions. So here we're really conceptualizing race as a social factor, and that the epigenome may be a biological sensor of cumulative chemical and non-chemical stressors. Clearly, these findings need to be validated. There are some substantial limitations. Both were cross-sectional studies with small sample sizes. We only had a single urine sample, which can lead to exposure misclassification. In the first study, we extracted information about fibroid characteristics from pre-existing ultrasounds or MRIs, so there's some heterogeneity. And, in terms of the interpretation of the modification by race, ethnicity in the epigenetic study, there is some speculation about what that means because we did not include measures of racism or social stressors. I want to end by talking about how we're moving the work forward now with kind of Forge 2.0 using a more explicit intersectionality framework. We've actually started integrating sexual and gender minorities. So we are now recruiting trans men into our population. They have been a largely ignored population in gynecologic studies and in environmental health studies. We are focusing on identifying novel epigenetic signaling pathways in fibroids and have some funding from NICHD to do that work. And we're also characterizing impacts of gendered racism using innovative mixed methods approaches. So just want to talk a little bit what we're doing on the epigenetics front. So in the kind of proof of concept study, we were really, we had to recruit women undergoing surgery, collect their fibroid tissues to look at microRNA signaling. Here we're trying to examine the utility of extracting exosomes or extracellular vesicles from blood in women with fibroids to see if we can capture information from fibroid cells that is being released into circulation, into the exosome. So we're trying to essentially identify a non-invasive biomarker of uterine fibroids, as has been done for certain types of cancers using extracellular vesicles. On the whole other side of things, we're also advancing how we're measuring and evaluating gendered racism. We just published our first qualitative paper from this study looking at Black women's psychosocial experiences with fibroid diagnosis and treatment. We're now integrating validated measures of everyday racism and gendered racism, including the gendered racial microaggression scale, which captures experiences of racism and sexism simultaneously. And we're conducting more qualitative work to understand how gendered racism manifests in our study population. So with that, I'd like to thank my co-investigators, all the research assistants at GW, past and present, as well as kind of the startup funding that I had to kind of start this work, as well as the funding that I have now to kind of grow and maintain it. Thank you.
Great. Our next talk is from Dr. Anna POLLAK, whose research focuses on the relationship between environmental chemical exposures and fertility, pregnancy, and gynecologic health. Dr. Pollak's research also addresses disparities in exposure, which stem from both environmental and occupational sources. Prior to joining George Mason University in 2013, Dr. Pollak was trained in reproductive and environmental epidemiology and epidemiologic methods. She received her PhD in epidemiology from Johns Hopkins University, her MPH in environmental and occupational health from George Washington University, and a BA in biology from Smith College. She completed a postdoctoral fellowship at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. So our second video will be from Dr. Anna POLLAK.
Hi, and thanks so much for that introduction. So my talk is going to be on non-persistent chemicals in relation to uterine leiomyoma, findings from the ENDO study. I'm Anna POLLAK. So uterine fibroids, as you just heard, are non-cancerous tumors that grow in and around the uterus. They confer an estimated cost of between $6 to $34 billion a year, and that is combined direct medical expenses as well as lost productivity. And they have a lifetime prevalence of between 70 to 80% in women of reproductive age. There's few well-established risk factors for uterine fibroids, but the most clearly established thus far are race, including Black race, and age, being of reproductive age. And so because of that, we sought to understand whether some endocrine disrupting chemical exposures may be linked with uterine fibroids in a cohort of women recruited in the ENDO study. And so we were interested specifically in 3 groups of non-persistent chemicals, which are widely found throughout different consumer products. These included bisphenol A, which is found in polycarbonate plastics, as well as epoxy resins of can coatings and cash register receipt paper. BPA is an estrogenic chemical, and so it's thought that it may exacerbate or augment the body's own endogenous estrogens, thereby promoting the growth of uterine fibroids, being a pro-estrogen. And ultraviolet filters, we looked at benzophenones and their metabolites. These are chemicals that are found in sunscreens as well as other personal care products, and they're also thought to be estrogenic. Phthalates are a group of chemicals that are found in plastics, fragrances, and personal care products, and some of these have been found to be anti-androgenic. And so the research question that we set out to answer in this study was, are spot samples of phthalate metabolites, bisphenol A, and ultraviolet filters measured prior to pelvic surgery associated with uterine fibroids? And so over here on the right, you can see kind of everything is fairly cross-sectional. Although the BPA, ultraviolet filters, and phthalate metabolites were measured just prior to pelvic surgery, that wasn't, the lag really wasn't long enough for those fibroids to have grown and developed. So that is what we were focusing on in this study. And the design was such that recruitment occurred at diagnostic or therapeutic laparoscopy or laparotomy in either Salt Lake City, which recruited 431 participants, or San Francisco, which recruited 63 participants. And the study took place between 2007 and 2009. The inclusion criteria were fairly broad: women who were aged between 18 to 44, and who were currently menstruating. The exclusion criteria included a history of laparoscopy confirmed endometriosis diagnosis, breastfeeding within the last 6 months, use of injectable hormones within 2 years, or history of non-melanoma skin cancer. And in terms of the methods, data collection occurred prior to that diagnostic or therapeutic surgery. And there was a single non-fasting urine specimen taken at the data collection time point. Study staff were trained in order for anthropometry measurements to be completed, and a computer-assisted interview took place. In terms of our statistical methods, we used logistic regression to evaluate the odds of fibroids, which were diagnosed surgically, or no presence of fibroids. And in our models, we adjusted for age, race, body mass index, smoking using established urinary creatinine cut points, and urinary creatinine, as well as study site. The chemicals were modeled separately, and the chemicals were log transformed due to their extreme right skew. We additionally conducted a sensitivity analysis wherein our comparison group was restricted to women with no other gynecologic pathology. And so since the study recruitment framework was that women were experiencing pelvic surgery, and that was part of the recruitment process, this sensitivity analysis enabled us to restrict only to those with a clean pelvis. So looking at fibroids versus no gynecologic pathology at all. And then an additional sensitivity analysis, we restricted only to women with fibroids only. So that was only 63 women, and that excluded the 36 women who had, upon surgical confirmation, both uterine fibroids and endometriosis. So while I think there may be a shared etiology, this enabled us to get at whether or not there's a, in looking at this separately, whether or not that contributed to bias in our findings. And so in terms of our study characteristics, women with fibroids were older, about on average 38 years old, compared to those without fibroids, an average age of 32 years. And there were no differences in body mass index between the two groups. In terms of women who had ever been pregnant and had a live birth, half of the women with fibroids had, and 59% of the women without fibroids had. Some additional study characteristics, as we would expect, a lower percentage of women with fibroids were white compared to those without fibroids. So 62% of those with fibroids were white compared to 78% without, and Black, Asian, multiracial, or other race made up 23% of those with fibroids and 9% of those without. In terms of the indication for surgery, women who ended up being diagnosed with fibroids upon surgery, about half of those women, fibroids was the indication for their surgery. And pelvic pain was the indication for about half of the women without fibroids. So those were kind of the leading surgical indications. And in terms of exposure levels, I'll start off, I know this is a bit small, but for phthalates, all of the phthalate metabolites were really quite similar across women with and without uterine fibroids. So here we're showing the geometric mean and 95% confidence interval. The exceptions were for bisphenol A and for the benzophenone derivatives 4-OHBP and 2-OH4-MeO-BP. Those were all found to be higher in women with fibroids as compared to women without fibroids in terms of the geometric mean. So that's only looking at exposures, not modeling anything, just geometric means. And then we went on to run our logistic regression models. And from that, what we found was that there were essentially no associations between any of these chemicals and uterine fibroids in any of our modeling approaches, including in our sensitivity models. And so, we recognize that this work was subject to some limitations. It was a spot urine sample, which these chemicals are quite short-lived. They're non-persistent. And so while some studies have found that one spot sample does kind of fairly well characterize exposure for some of these metabolites or some of these chemicals, for others, there's really so much day-to-day variation that one spot sample may not be adequate to characterize exposure. And then for surgical recruitment, these women, this study population may not be generalizable to those who were, not seeking to undergo laparoscopy in order to address gynecologic or other pain. And then the study population overall was fairly white and lacked diversity, since we know that uterine fibroids are more prevalent in Black women. I think that was a limitation, that the study population did not include many Black women. And so kind of the ideal way to measure this, if it were up to me how to ask this question, would be to look at these chemical exposures several times during pregnancy, and then again look at these exposures when the children were young, were perhaps going through mini puberty and pubertal development, and then follow the women forward for tracking development of uterine fibroids. There's some evidence that supports that uterine fibroids may in fact have an in utero developmental origin, kind of while the organs are in development, and that those exposures at those times may reflect critical windows, and so that would enable the analysis of that question. I'd like to acknowledge my co-authors and collaborators, especially Jermaine Buck-Lewis and Carintha Chalam-Cannon at George Mason and NYU, respectively, as well as my other co-authors. I'd like to acknowledge funding for the ENDO study from the NICHD and my own funding from NIEHS. Thank you.
Excellent. So our third speaker is Dr. Amelia WESSELINK, who is a research assistant professor in the Department of Epidemiology at the Boston University School of Public Health. Her research focuses on how environmental exposures influence reproductive outcomes, including fertility, miscarriage, and benign gynecological disease. She earned her PhD from BUSPH, also in the Department of Epidemiology, in 2018.
Hello, everyone. Thank you so much for joining this webinar. I'm really excited to be here today to share with you some of the research my team has been doing on exposure to endocrine disrupting chemicals and risk of fibroids in the study of environment, lifestyle, and fibroids, or SELF, which is a prospective ultrasound-based cohort study of reproductive aged Black women. Hopefully by this point, you don't need additional convincing that fibroids are a clinically significant and understudied health condition, and that it is biologically plausible that exposure to EDCs could influence fibroid risk. So I'm going to start my talk by highlighting 3 key gaps in the epidemiologic literature on this topic that motivated our work in SELF. First, most prior studies have been either cross-sectional or case control studies, and have thus measured exposure to EDCs either at the time of or even after case diagnosis. So these studies are really missing out on the etiologically relevant window of exposure. Second, almost all studies have relied on self-report or hospital or clinical records for case ascertainment. And because of the high proportion of asymptomatic cases, this method of case ascertainment likely has quite low sensitivity. And because a clinical diagnosis relies not just on severity of symptoms, but also on access to medical care and willingness to seek care, the potential for differential outcome misclassification is high when exposure is related to these factors. And finally, most of the studies on this topic have been conducted primarily in white and Asian populations. And there's been limited study of exposure to endocrine disrupting chemicals and fibroid risks in Black women. And this is really important for a couple of reasons. First, fibroids is a disease for which there is a known racial disparity. Black women have higher incidence of fibroids, earlier onset of symptoms, and more severe symptoms compared with white women. Established risk factors, socioeconomic status, and differential screening or medical care do not explain this disparity. And EDCs may actually be important contributors to racial disparities in fibroids. We know that EDC exposure is not equitably distributed across populations in the United States. This figure, which is not intended to be exhaustive, illustrates a few mechanisms through which historical and contemporary racism and oppression have led to inequitable EDC exposure by race. A key example that is of particular relevance to Black women is EDC exposure through personal care product use. Racial discrimination based on skin tone and hair texture, racially targeted advertising of specific beauty products, and perpetuation of European beauty norms have led to increased use of specific personal care products among Black women. And I highlight this fact to remind you of the social and historical context of these exposures, and to emphasize that this context is really important when we're talking about racial health disparities. So to fill some of these gaps in the literature, we examined the association between biomarkers of EDC exposure and fibroid risk and growth in the SELF study, which is a prospective cohort study of Black women from the Detroit, Michigan metropolitan area. Recruitment and enrollment took place during 2010 to 2012. Overall, we enrolled 1,693 women who were aged 23 to 34 years, who self-identified as Black or African American, who had an intact uterus, and who had no history of fibroids, cancer, or autoimmune disease that required treatment. We followed women up every 20 months for up to 80 months, and the 80-month visit is currently ongoing. This table shows an overview of the type of data we have collected at each time point. At these visits, women undergo a transvaginal ultrasound, they provide urine and blood specimens, and they undergo an anthropometric examination. They also complete telephone and computer-assisted questionnaires, a 9-day menstrual diary, and a questionnaire on use of personal care and consumer products during the past 24 hours. The key advantage of SELF over other studies on fibroids is that we serially screen an entire population of women at baseline and prospectively over time to identify fibroids. We used transvaginal ultrasound, which has both high sensitivity and specificity to detect fibroids relative to histologic evidence. Histologic evidence is considered the gold standard, but is of course highly invasive. So by using this method, we were able to, first of all, exclude women with prevalent fibroids at baseline from our prospective analyses. We were able to identify new cases regardless of symptoms. We were able to assess fibroid characteristics like number, size, and location. And we were able to measure how fibroids grow and change over time. To maximize cost efficiency, we conducted a case cohort study within SELF. So 385 SELF participants had fibroids that were identified at the time of the baseline ultrasound, and they were excluded from the present analysis. From among the women who were fibroid-free at baseline, we selected a random sample of 592 women to represent the exposure distribution in the source population. We then identified all incident cases of fibroids that occurred during 60 months of follow-up, which was 310 cases. And some of those cases were to women who were part of the random subcohort, and some of them were to women who were not part of that subcohort. And so in the end, we analyzed EDC concentrations in urine and blood from the women in orange only. So women who were either part of the random subcohort or who developed fibroids during follow-up. And so our final sample size was 754. This slide provides an overview of all of the different chemicals we measured in this study. The chemicals in the left-hand column were measured in plasma, and the chemicals in the right-hand column were measured in urine. And in this talk, I'm going to focus on our results for phenols, triclocarbon, and parabens. But I'll mention some of our findings from these other classes of chemicals as well. For chemicals that were measured in plasma, which tend to be more persistent, we measured chemical concentrations in plasma collected at baseline only. For the chemicals that were measured in urine, which tend to be less persistent, we measured concentrations at each time point up until the time point before fibroids were detected. And from these concentrations, we calculated a cumulative average concentration for each risk interval. So for the risk interval from baseline to 20 months, we used baseline concentrations. For the risk interval from 20 to 40 months, we averaged baseline and 20-month concentrations. And for the risk interval from 20 to 40 months, we averaged baseline, 20-month, and 40-month concentrations. For analysis of fibroid incidence, we conducted a time-to-event analysis among women without prevalent fibroids at baseline. Women were censored at hysterectomy, loss to follow-up, or the 60-month visit, whichever came first. To measure fibroid growth, we identified matched fibroids, and that is fibroids that were identified on subsequent ultrasounds as being the same fibroid. So there's an example in the figure here of this fibroid at visit 1, which at visit 2 had grown but was identified as the same fibroid. And we measured growth as the change in log volume scaled to 18 months. We used a linear mixed model to assess the association between chemical exposures and fibroid growth, using data from 161 fibroids among 124 women. So now I'm going to go through some of our findings so far. Approximately 24% of our participants developed fibroids by the 60-month study visit. Most women who were diagnosed had fibroids that were small, and most also had just 1 or 2 fibroids, although the number ranged from 1 to 5 at the time of diagnosis. The most common type of fibroid was intramural, which are these tumors that are completely located within the muscular layer of the uterus. The next most common type were subserosal, which are these tumors that develop in the outer wall of the uterus. And then the least common type were submucosal, which are these tumors that develop in the endometrial wall and protrude into the uterine cavity. And although these were the least common, these are the type of fibroids that are most likely to cause severe symptoms. In terms of exposure, urinary concentrations of phenols and parabens in self were very similar to concentrations among reproductive age Black women from NHANES. This figure shows hazard ratios for a 1 standard deviation increase in exposure to phenols, triclocarbon, and parabens. And contrary to our hypothesis, these associations are largely null or weakly inverse. So for example, a 1 standard deviation increase in urinary BPS concentration was associated with a 7% lower rate of fibroids. One thing in particular to note about the parabens, which are at the bottom of this figure here, is that the longer the alkyl chain on the paraben, the more estrogenic activity it has. And so of the parabens we measured, butyl paraben is the most estrogenic, followed by propyl, then ethyl, then methyl. But what we see is that regardless of estrogenic activity, there's not really much going on here. This figure shows the percent change in volume for a 1 standard deviation increase in exposure. And we see some weak associations in both directions. So BPS was associated with positive growth, whereas 2,4-dichlorophenol and triclocarbon were both associated with negative growth or shrinkage. But it's important to note that across the board, these associations are very weak, so less than a 5% change in volume over an 18-month period. As I mentioned previously, we've looked at several other classes of EDCs in relation to fibroid risk in self. So we've published on polychlorinated biphenyls, brominated flame retardants, and organochlorine pesticides, and phthalates and phthalate alternatives. And across the board, the story has been pretty similar. We see little evidence of an association between biomarkers of exposure to EDCs and fibroid risk, and for specific chemicals, we're even seeing some inverse associations. So there are a couple of potential explanations for these findings. First, if the slight inverse associations are causal, it might be that these specific EDCs act in an anti-estrogenic manner in the uterus. And there is some precedent for this hypothesis in the literature. A study of women who were highly exposed to dioxin, which is a really potent EDC, after an industrial accident in Seveso, Italy, had 40% lower prevalence of fibroids 20 years later. And the authors of this study posited that it may be because dioxin has an anti-estrogenic activity in the uterus. Another explanation is that there might not be an effect of EDCs on fibroid incidence at low doses. Women in this cohort had relatively low exposures, and we aren't able to generalize to women with higher exposures using our data. And finally, it might be that EDC concentrations measured in adulthood are not relevant for fibroid risk. Our study had a couple of key limitations worth mentioning. So first, we measured EDCs at baseline and every 20 months. But the non-persistent chemicals have really short half-lives. And so measurement in a single urine sample every 20 months may not reflect regular exposure. So there's likely some misclassification happening there. Second, we measured EDC concentrations in biomarkers collected during adulthood. We did not have measures of exposure during critical windows of development in early life, including in utero and perimenarcheally. So if fibroid initiation actually begins earlier on, we will not be able to capture that with these exposure measurements. Next, we measured biomarkers of exposure to EDCs in the most readily available biospecimens, that is, urine and blood. But it's unclear, both from our study or from the literature, how urinary and plasma concentrations translate into concentrations in uterine tissue, which is presumably of greatest relevance, but logistically very challenging to obtain. And finally, we conducted ultrasounds every 20 months. And so we weren't able to identify the precise timing of tumor initiation. And again, this makes it really challenging to identify the appropriate exposure window. I want to end by thanking all of the collaborators that I have the tremendous privilege of working with on this project. Specifically Lauren Wise, who is the PI of our work on EDCs and fibroids in self, Olivia Orta and Victoria Frew, who led the work on some of the other classes of chemicals in relation to fibroids, Birgit Klaus Henn, who's leading our work on chemical mixtures and who you will hear from next during this webinar, and Donna Baird, who is the PI of the self-study overall. I'd also like to acknowledge the team at Social and Scientific Systems who helped develop and manage the study, as well as the clinical staff at the Henry Ford Health System who ran all of the study visits. And of course, a big thank you to the NIEHS for funding this work and to all of the self participants for their tremendous contributions to this research. Thank you so much, and I'm happy to take questions at the end of the webinar.
So our next speaker, our final speaker, will be Dr. Birgit Klaus Henn, who is an associate professor in the Department of Environmental Health at Boston University School of Public Health. She has over 15 years of experience in environmental health and epidemiology with expertise in chemical mixtures, endocrine disrupting chemicals, and pediatric health. Dr. Klaus Henn has examined associations between environmental exposures and neurodevelopment in multiple pediatric cohorts worldwide, spanning critical periods of development from prenatal exposure through adolescence. Dr. Klaus Henn received her PsyD in environmental epidemiology from Harvard School of Public Health and her MPH MBA from the University of California, Berkeley.
Hi, everyone. Thank you for the introduction, and thank you to the webinar organizers for this opportunity to present. So I'm going to build on what my colleagues have already very nicely laid out in terms of endocrine disrupting chemicals and risk of uterine fibroids, but my talk will focus on mixtures of EDCs. I hope to do three things in this webinar. I'll describe a few approaches for analyzing EDC mixtures data. I'll show some preliminary analyses using these approaches in the self cohort, and I'll finish with a few thoughts on future directions. So we already heard from previous speakers that EDCs are all around us. They occur in many common consumer and personal care products, packaged foods, building materials, and so forth. And these EDCs represent several chemical classes as depicted here. So these exposures are commonplace and are happening more or less continuously. We also all know very well that these EDC exposures do not occur as individual chemicals, but rather they occur as a complex mixture that includes simultaneous and sequential exposure to multiple EDCs. But the motivation for this work goes beyond the fact that EDCs are co-occurring ubiquitous exposures. And I'd suggest that for fibroids, there's particular relevance in examining EDC exposures as mixtures for a few reasons. So first, we heard from previous speakers that there are few modifiable risk factors that have been identified for UL. And despite the biologic plausibility for links between EDCs and fibroid risk, few human studies have demonstrated an association so far. Certainly there are methodological limitations in some studies, but it is plausible that we are missing the true impact of EDCs on UL because we've mostly been looking at one EDC at a time. Second, we just heard that one possible explanation for the lack of individual EDC associations with fibroids is that exposure levels are just too low. There is, in fact, some evidence, though, that at least in vitro experiments, even if exposure to an individual EDC is too low to see any impact, multiple EDCs can work together to exert an effect. And last, EDCs seem to interact with hormone receptors in unusual ways. Individual EDCs may interact with multiple different hormone receptors simultaneously, and multiple different EDCs can interact with the same receptor. So if we're exposed to multiple EDCs as a mixture, which we likely are, then we have a cocktail of mixed interactions with receptors that might be competitive, or they might be additive or more than additive. So there's an opportunity for there to be complex biologic interactions between EDCs. Yet, there are sparse epidemiologic data on EDC mixtures and risk of fibroids. To begin to examine EDC mixtures, or really any mixtures, a good starting place is to ask ourselves, what are the key questions that we want to address? And typically there are 4 questions of greatest interest to epidemiologists for addressing mixtures. First, what are the health effects of the individual EDCs in the mixture? Second, are there interactions between EDCs in a mixture? Third, what is the total effect of the EDC mixture as a whole? And fourth, are there particular patterns of exposure in different populations that can either help us prioritize which mixtures to focus on for evaluating health impacts, or to help us target interventions to reduce exposure? So identifying our questions of interest helps guide our choice of analytic method, because different analytic approaches, different statistical methods tend to answer different questions. So the number of approaches that are now available for analyzing mixtures data has been growing very quickly over the last decade or so. And we now have a fairly lengthy menu of approaches that could be useful for your mixtures data analysis. So here's one way to organize these various approaches. And this graphic I borrowed from my kind colleagues at Columbia. This shows some of the, I would say, more commonly used mixtures methods here in the smaller bubbles. And we can group these approaches roughly by those 4 questions I showed you in the previous slide, here in the larger bubbles in the center. So this is how our questions map onto this graphic. This is not, of course, an exhaustive list of mixtures methods, but it can help guide our choice. The other thing to note is that there are other considerations when deciding on an analytic approach, such as the type of variable that you have, your sample size, number of exposures, and so forth. So I'm going to shift now to showing some examples from our work in the SELF cohort, which Amelia introduced in her previous talk. As a reminder, this is an ongoing prospective cohort of Black women in Detroit, and these women were free of fibroids at baseline, as determined by transvaginal ultrasound, and were then followed up at 20-month intervals at 3 follow-up visits. We estimated exposure to several classes of EDCs, shown here, both non-persistent and persistent. And for the analyses I'll show today, we had about 750 participants. So we're really interested in, well, really in all of the mixtures questions I laid out previously, but we considered these in 2 parts. In the first part, we want to estimate the individual joint and interactive effects of EDCs on fibroids. And in the second part, we want to describe patterns of EDC exposure in our participants. So to address the first part about individual joint and interactive effects, we chose to use Bayesian kernel machine regression, or BKMR. And many of you have probably some familiarity with this approach, but briefly, BKMR is a technique to estimate the joint effects of multiple correlated exposures. It's nice because it's highly flexible and that it allows for nonlinearities between the exposures and the outcome, and it allows for interactions between exposure variables. And that includes higher order interactions. So with this method, our health outcome, represented by Y in this equation, is regressed on a flexible function, the H function, that represents the association between a set of exposure variables, here are the X's, and the health outcome adjusted for confounders. So with this approach, we can also get information on which of these exposures in the H function are important predictors of the health outcome. And in the end, we have 2 types of output. We have a smoothed high-dimensional exposure response surface that I like to think about as a multi-dimensional smoothed plot. And we also get posterior inclusion probabilities for each exposure variable in that H function, which is essentially a relative ranking of variable importance. So what I'm showing here is a general framework, but BKMR can be applied to binary outcome data, which is what we have with our fibroids data. We chose BKMR because of its ability to address all of our research questions of interest in terms of joint interactive and individual associations, but also to allow for possible nonlinearities and to allow for different EDCs to act in different directions on the outcome. So we applied BKMR to our self-data on non-persistent EDCs, and this is work led by Doctors Victoria Frew and Amelia Wesselink. I just mentioned the smooth exposure response surface that's estimated with BKMR, and so from that surface, we can pull off various estimates or contrasts. So here in this figure, we've plotted the difference in risk of UL when concentrations of all EDCs are at a given percentile, so here noted on the x-axis, compared to when all EDCs are set to their median, are at their median. So what we're seeing here, what this suggests is that compared to when all EDCs are at their median here in the center, UL risk is higher when EDCs are at lower levels, at lower percentiles. So what's driving this? Which EDCs are important? So we can look here at the exposure response curves for each EDC in the mixture. So these panels represent associations between each individual EDC and UL risk when all the other EDCs are fixed at their median. And we can see a few things here. Associations in both we see associations in both positive and negative directions. So not all EDCs are acting in the same direction. There's a little evidence for nonlinearity for a few of these EDCs. And then based on the inclusion probabilities, these 6 EDCs highlighted in red had the highest rankings of relative importance. So you can see that most of these associations for the highest ranking EDCs are negative, which is consistent with the overall mixture effect I showed in the previous slide. We did also look at interactions and saw some evidence that associations for MECPP, a metabolite of the phthalate DEHP, varied at different levels of the mixture. And we're looking into this further. So as a comparison, we were interested in using a method that is less computationally demanding and may have some benefits in terms of interpretability. So very briefly, quantile-based G computation is based on developing a new exposure variable, an exposure index that is a weighted average of all exposures and represents the mixture as a whole. And this new index variable is then used to estimate associations with a health outcome. So the output of this approach is a regression model between the outcome and this new index variable, as well as weights for each exposure in the index that represent variable importance. And these weights can either be positive or negative, reflecting the direction of their associations with the outcome. When we applied QG-comp to our self-data on non-persistent EDCs, we saw, similar to our BKMR findings, that different EDCs act in different directions on the outcome. And you can see that here reflected by the positive and negative weights. We also saw that the top weighted EDCs identified by QG-comp are similar to those identified by BKMR as having the highest inclusion probabilities and are in the same direction as BKMR. So there are some consistent findings here across methods. So the last question we wanted to address was about identifying patterns of EDC exposure. And there are a couple of reasons why we wanted to do this. First, with a better understanding of patterns or profiles of exposure, we can identify factors that are linked with those particular exposure profiles and then develop more effective targeted interventions for reducing exposures. And second, we can use these exposure profiles in subsequent mixtures models with the health outcome. And the advantage there is that we have fewer dimensions without a loss of information on exposures. So there are, again, a number of ways we could identify patterns or profiles of exposure. Principal component analysis, or PCA, is just one possible approach. This is a dimension reduction technique that is fairly well known now in the field. But the concept is that our large number of exposure variables that may be correlated can be transformed into principal components, new variables, essentially, which are now uncorrelated. So we applied this approach to self-data on 33 persistent EDCs from 4 chemical classes. And this is work done by Samantha Schildroth, one of our doctoral students. We found that 4 principal components explained about 75% of the variance in EDC concentrations. And we describe these profiles of exposure in terms of their composition of EDCs, shown here in the 4 groups. We also found that correlates of these profiles included various dietary and behavioral factors, such as smoking, BMI, and poultry intake. So to summarize, based on these very preliminary analyses in self, increasing levels of the non-persistent EDC mixture did not appear to be associated with increased UL risk. In contrast, our findings suggested that lower levels of the mixture might be associated with higher risk. So we used a few different approaches to address several mixtures questions. And I think it's important to note that every statistical approach has pros and cons. So today I talked about chemical exposures exclusively, but of course there are many non-chemical factors that also come into play. And as we move more toward exposomics, we must include intersectionality and consider how racism, sexism, and related concepts intersect with exposure to EDCs and health outcomes. In future work, I think it could be useful to consider other metrics, such as symptom severity or age at onset. And importantly, we should consider timing of EDC exposures, because exposures in earlier life or during critical developmental periods may set us on a particular trajectory for fibroid development in adulthood. And we may not be capturing those critical exposure periods in studies that we've conducted to date. I'd like to acknowledge all of my colleagues on the self-study, in particular Lauren Wise and Donna Baird, and the colleagues and collaborators shown here. I'd also like to thank the study participants and funding from NIEHS. And finally, thanks to you all for listening, and I'm happy to try to address any questions.
Great. We've had 4 outstanding talks. And if all of the speakers could turn their cameras on, we can start having some discussion. Those were all really wonderful talks, and it was great how there's just so much synergy there in the work that's being done. So I think to start, we have a couple of clarification questions from the Q&A. And so I think the first one I will send to Anna, because I think it's relevant to your R1. So it's from Bridget York asking, will you also be researching endometriosis as we find more and more women being diagnosed with endometriosis after fibroid morcellation surgery, both in and outside the womb, with or without disseminated fibroids, and may be inatrogenic after morcellating the fibroids, and the cellular material may be very similar?
Yeah, that's a great question. So the first thing that comes to mind is maybe AMIs or self are kind of set up in order to look at that, since they're following individuals post surgery. But yeah, I mean, I think it's a really important point considering endo as well, as definitely that's another outcome that I am really interested in looking at. And one of the things that I've been thinking on for a while is that the importance of considering structural racism and, I guess, intersectionality, as Ami so nicely framed it earlier in the webinar, affects the diagnosis of endometriosis. So I think that is an area that I'm hoping to look at in the future as well.
Anything to add from the other speakers on endometriosis?
I would just say in our study, we are definitely collecting careful information on other gynecologic comorbidities. And that, we're kind of, because of our study design, able to kind of do this through pathology reports from surgery. And we're finding that like 50% of the women, or I don't know, between 30 to 50% of the women with fibroids have undiagnosed endometriosis or adenomyosis. It's very, very high. And so, I think there are some interesting questions to ask around kind of, these, I think in a lot of cases, these are not independent conditions, right? And so what are kind of underlying what are underlying biological mechanisms that may be impacted?
Okay. So another question, I'm trying to manage this a little bit. So there's a question about one of the talks. I think this one might overlap with either Anna or Amelia. The question was, did the study allow for the presence of IUDs, copper or plastic, and or the presence of plastic surgical mesh? I think it's about like the exclusion criteria.
That was not an exclusion criteria as long as it was not hormonal. A hormonal IUD, I believe, would have been an exclusion criteria for endo.
And in SELF, we did not make exclusions based on hormonal contraception, but we did collect information on it and we controlled for it in all of our analyses.
So another question is, to what extent do you think variability in fibroid characteristics explains some of the differences in findings across the study?
Well, I'll bite. That's okay. So, I mean, I think kind of the framework from which individuals were selected is pretty different across, like FORGE, right? Is Ami your acronym? Mm-hmm. FORGE, SELF, and endo. In that FORGE and endo, individuals are presenting for surgery, whereas in SELF, you're getting that prospective growth, which I think is a really key aspect. Although I thought we had I was surprised actually by the similarities between endo and SELF in terms of kind of the null findings for phthalates. And so I'm not, even though the recruitment approach was more similar between FORGE and endo, I would have expected like whatever selection forces, drove that to lead us to have more similar findings when what Dr. ZOTA was finding was some positive associations with phthalates. And so, yeah, I'm not sure why that didn't work out the way I would have expected.
Well, I think too, right? I mean, your two studies were looking at incidence or risk, right? Whereas we were kind of, our study was a kind of more of a case only study. So looking more at, what we think are clinical measures of severity or kind of fibroid characteristics. So kind of asking, kind of different questions. I think, and I think the other thing, right, is that I think all the points that have been raised are really important. And I think the SELF study is, a gold standard study, like so well designed to ask really, really unique questions. I think, right, still at this point in the study, most of the fibroids are still pretty small too, right? And they haven't exhibited a lot of growth. And there is kind of this interesting thing where there is some signal in the kind of experimental literature, I think. And there was a really interesting study very recently that specifically looked at kind of certain phthalates and kind of measures of growth and even uterine volume, like in a experimental setting. And we thought that was really interesting. But then, right, the signal in a lot of the epi studies is not there. So there's something that's, not potentially being transferable, or is it, whether it's not the right outcome measures or the timing of exposure or so many important issues have been brought to light here.
Another thing to add about fibroid is I haven't seen this for endocrine disrupting chemicals, but I know there's been work on smoking that has shown that different fibroid subtypes, so whether it's submucosal or subserosal or one of the other types, potentially have different risk factors. So that's another thing to consider moving forward that we haven't really taken into account here.
And since you brought up, Ami, the issue of timing of exposure, and all of the speakers touched on this. So I think one of the questions is that, is there direct evidence supporting the idea about kind of critical windows of exposure that predate diagnosis, or is it more of a theory for developing more human-based or animal model research?
So I can start this if that's okay, unless you want to go. But yeah, no, I think that there is some evidence early in animals that rodents who are developmentally exposed, or in those animals, fibroids appear later in life at a higher frequency, and I think certainly most hormones don't act at all times, right, during the life cycle. So because receptors are only found at certain times during development. So I think there are some examples, and even in humans, exposure to DES. So there are some examples I think that we can draw from that justifies, I think, the need to look at critical period forward in time, right, to look at earlier life stages and exposure during earlier life stages.
I think another potential sensitive window that would be, interesting to think about in the context of fibroids is pregnancy, like for the women themselves, just because, right, it's a big time of hormonal fluctuation. We actually know that fibroids can change during pregnancy. They can often get smaller either during pregnancy or in the postpartum period. And so, right, I think that's a kind of a growing trend overall, to think about kind of pregnancy as a sensitive window for women's health kind of across the life course. And so that could be another interesting window to explore beyond kind of the developmental window, I think.
And thinking about developing studies related to generational exposures or developmental windows, and Birgit, you mentioned DES, where we have these long cohorts, and Amelia, you mentioned Sebastian. And, do you think there's a way to move forward? One of the questions is, do you think there's a path to move forward to look at these periods, given the age at which fibroids tend to become apparent, right? So you have a little bit of an older population that has symptom severity that may lead to surgery, compared to some of what's being detected asymptomatically in smaller tumors, benign tumors in self. So, what are some approaches that you think we may be able to use now or may need to develop to do this work?
Sure, yeah, it's a great question. I think, as Ami alluded to, if we could even just start among sort of preconception, among sort of pre-pregnancy cohorts, that's one place to start. But even moving to younger ages, I think would be really interesting, looking at adolescents or pre-adolescents. And if there's a way to, leverage some longer term prospective cohorts, I think that would be my first place to look, ideally, if that's possible. Obviously, there are some other limitations there in terms of cost and feasibility, but that would be ideal.
Yeah, I think I totally agree with Birgit. Going back and utilizing studies that have already characterized prenatal exposures to kids or to infants who are now grown up, and then adding in some sort of gynecologic health endpoints of interest to those studies. Yeah, I think it's just an important component. And I think one of the things that I try to advocate for among my students is to remind them that, like, women's health matters more than just creating the next generation of people. Like, we matter for ourselves. And so that's one of the things. And then I wanted to touch on, Tracy, one of the things that you were saying of treating all fibroids as fibroids. I think it was at SPER last summer. It was really in December, but it just happened. That I was listening to a talk thinking about endo as and thinking about it in the same way as breast cancer, and that we're not quite fully there yet, at least with respect to our epi methods for endometriosis or uterine fibroids. And so that may contribute to some of kind of, if we're mixing together our outcomes and they're really driven by different underlying mechanisms, then we need to get better about that, or at least find out if that's the case.
Yeah, I just, to kind of expand on what Birgit and Anna said, I mean, I think a couple of things. I think the fact that self-report of fibroid outcomes has high measurement error, I think makes it difficult to sometimes kind of add these outcomes into kind of large cohort studies, because, right, obviously the gold standard way would do repeated ultrasounds like Self is doing, but that is a big ask and may be beyond the capacity of a lot of studies. And I mean, that's one reason we're doing this kind of epigenetic biomarker work, is like, it's kind of high risk, high reward. But if we can find kind of easier biological markers to kind of get at underlying activity, that, it could open up a lot of pathways, new directions in epi to study fibroids and maybe some other gynecologic conditions, because I think what the big roadblocks is just that these conditions are so hard to study well in a population health setting. And so that's one reason we're kind of pursuing the kind of exosomes, extracellular vesicles avenue. And around the kind of diversity within kind of what we call fibroids, I presented at the NIEHS fibroid biology meeting. I guess it was last February that Donna Baird helped to organize. And it was largely like basic scientists and clinician scientists. And I was like one of the only population health people. That was really one of the takeaways, that like no two fibroids are the same, and there are all these subtypes, and they got way into that kind of stuff, and they just wanted to measure more and more things at the molecular level. And so, I think there's like a trade-off between like getting too specific and, and creating measurement error in your outcome. But, to your point, that was definitely kind of one of the takeaways for me at that meeting, was that there's so much variability within this kind of umbrella of thing we call fibroids.
And I think that relates to one of the comments in the Q&A box, is that another variable to consider is adenomyosis, which can confound measures of uterine volume, and can also be a source of comorbidity among women with uterine fibroids. And our time is coming a little bit short, but I would like to address, since we're talking a lot about, we've talked about intersectionality, we've talked about disparities. And so I was wondering if you could comment on how you approach this research, especially thinking about patterns of product use, or even where you talked about diet and behavioral factors, how you approach this research without stigmatizing the groups that are often underserved or have been affected by societal patterns out of their control.
I'm happy to start. I mean, we, our populations are majority Black women. We have been very intentional about having a diverse kind of staff of RAs. And overall, we have really high recruitment rates. And I think it's because, especially a lot of Black women, fibroids really have a big impact on their lives. They're just really so happy and appreciative that people are researching this. And in our case, we're kind of very explicit about kind of looking at social, environmental, molecular determinants. And so, that's part of our IRB. So I think they appreciate kind of the holistic lens. You know, sometimes like, you know, we'll get pushback. Like sometimes some of our participants will be like, I don't want to talk about that, or I'm not going to answer those questions. But I mean, I think kind of in all of the research I do, I'm increasingly trying to be very, very explicit about the social structural dimensions of environmental exposures, of product use, of where you live, right? So I think we have a lot of power in kind of the research questions we ask, how we, you know, how we translate those findings, and how we talk about them. And so making it less about kind of just proximate risk factors, right? And really shining a light on kind of the structural factors that shape our kind of environmental, occupational environments.
Excellent. So thank you everyone for joining us today. This has been amazing. So much great work and so many great new ideas and good discussion. I'd be remiss if I didn't also thank Joan and Maranati and also Lauren Wise and Simon, who has definitely been a huge help with the tech today. This has been great. Go forth, stay healthy, do great work, and thanks so much for joining us.